Hi everyone, welcome to Farsight's Biotech and Health Extension Salon series. I'm really delighted today to welcome Gizem Gumuskaya to our seminar series, and she'll be discussing synthetic morphogenesis, self-constructing living architectures by design. This is a seminar that was nominated by Michael Levin, and we're really so delighted to have you here. We've had Michael on for a few times now, and I think the work that you're doing is building up, or much of the work in collaboration with him in really fantastic ways. So we cannot wait to hear more about it. Please take it away.
Of course. Thanks so much for having me. We've been trying to schedule this for a while now. I'm so happy that we got it in the books and it's happening. All right. So hi, everyone. Again, I'm Dr. Gizem Gumuskaya, and my research for the last 10 years has been focusing on synthetic morphogenesis, which is this endeavor of trying to create self-constructing living architectures by design. As some of you might know, I actually started my career as an architect. I'm originally from Turkey, so I did a undergrad in architecture in Istanbul Technical University, and then came to MIT in 2015 as a innocent architect. Little did I know that shortly after coming to MIT, I was gonna, just being in Boston, meet with this field of synthetic biology. And what really captured my imagination as a designer in synthetic biology is this idea of now there's this paradigm shift in how we see nature, right? We're so conditioned to see nature, at least like traditionally, as this thing sitting outside, waiting to be investigated by scientists and understood and mapped. But now synthetic biology was saying that, no, it's actually an active design medium, and this is something we can actively design with. So that ended up me doing a dual master's at MIT, and then finally doing something I never thought I would as a designer, getting a PhD in biology. I did that at Tufts jointly with Harvard Wyss, and I'm so grateful in both institutions having found advisors. So at MIT, it's Ron Weiss, and at Tufts, it was Mike Levin, who really had the vision to be able to support this really ambitious, rather, goal of combining design and biology. I have some examples here. We'll dive more into what this looks like. But first of all, again, this wanted to go back to the beginning. Okay. I'm trying to advance my slides. Okay. I think it doesn't want me to keep laser pointer when doing that. But anyway, so this is again a background for why I found this super fascinating, because nature has this fabrication framework essentially that does not exist in anything that's been built by humans in the course of human civilization for the last 10,000 years. And specifically, this idea of having a seed being planted and that seed having all the information to build the final architecture, and also in the process being able to propagate itself, was really fascinating. And again, just to contrast it to this traditional way of manual construction in nature, we're all familiar, you don't need to be an architect to know this, we're all familiar with this traditional workflow. You source your material, you manually shape your material, and then you do this kind of assembly based on an external blueprint, and then eventually transport it to the site. Obviously, these two steps can switch based on the particular fabrication modality you're using, but this is really quite expensive, and not to mention, this is contributing to the 48% of the CO2 released to the atmosphere. So it has quite a big impact in global warming as well. And again, what really captured me in biology is this idea of, okay, if we have this magical substance, a biological cell, that can do all of this and more. It can create raw materials, so you don't need to go sourcing it. And not only can create it, it can also self-replicate it. Once this video reverts to the beginning, you'll see that this whole construction process started as a single cell, and then that cell started exponentially growing. And not only generating this raw material, but also in the process, organizing this material, constructing this material towards a functional shape. You really no longer need to give form to this material manually. It does it for you based on this embodied instructions, the embodied morphogenetic code. And based on that, all these amazing architectures we see in and around us emerge. And this, therefore, can actually happen in situ. You don't need to fabricate anything anywhere and transport it. Things can grow on site. And the reason why this is particularly exciting in 21st century is, for the first time, we actually have insights to how to edit this process. Obviously, we're all familiar with all of these things happening in nature just by being mere observers, but What's really happening now is that we are entering an era we can engineer this process of morphogenesis. Because the audience is mostly bioengineers, I'm going to go into a little bit more detail on like how we've been traditionally doing this. So using the word traditional in quotation marks, because synthetic biology as a field is really now like only 25 years old. But what really enables this approach, this engineering the process of morphogenesis, is our recognition as scientists that there is in fact like tremendous logic beneath this sort of seemingly magical process. So in the process of that single cell becoming exact organism, I believe zebrafish. So if there are any ecology people there, they can correct me, but I'm pretty sure it's a zebrafish. In the process of becoming a zebrafish, this organism, this multicellular structure, actually, this is what happens under the hood. And this is very reminiscent of our human-made technology. So if we just the computers and everything we're interacting with are running on these circuits, instead of though now creating these circuits with transistors and connecting them in different ways, we are now starting to, so you can create like PCBs and mess with these transistors and generate all kinds of complex behavior via Boolean logic. Instead of now doing that, we are asking the question in synthetic biology, can we do the same thing with genes? Can we connect these genes in entirely novel ways to create new behaviors in cells via Boolean logic that would not otherwise be possible? Specifically on the synthetic morphogenesis front. So we're using this approach, synthetic biology, for all kinds of things, drug delivery, creating new valuable molecules, just really on a lot of industries like energy, but specifically on the front of using this to create structures. There has been a lot of great advances. So I have a state-of-the-art summary here. So it started quite early, 2005, with like Romais showing that you can actually have two artificial cells, so like engineered cells, that have these circuits, and via interactions, they can actually create this sort of canonical developmental biology pattern, which is the bullseye. And then this was in bacteria early on. And then like bit more a decade later, and they were able to accomplish even more complicated patterns, in fact, a Turing pattern. And you can see like circuit is pretty crazy. And then also the system got moved to mammalian cells. So here we have the Mendel Limbs lab creating a similar 3-layer pattern, except this time with mammalian cells and then 3D. And then this got expanded with mammalian cells into much larger length scales once the diffusible morphogen technology was invented. These are all attempts to use this circuit-wise logic inherent in development to reprogram it towards new shapes. And just something from my own career. So this is where I started my journey of synthetic morphogenesis. And just to give you a bit more stepwise idea of how this would happen, you have your initial synthetic circuit that again has the genes wired up in a way that that drives the final morphology that you want to see. And then that single cell self-replicates, creates again that like self-replicating raw material, and it organizes into a spheroid in this particular case. So, but it's not just any spheroid. Thanks to the circuit and the synthetic morphogenetic algorithm it's driving, which I'll get into more in the next slide, we don't just have a spheroid here, we have a spheroid with actually 6 distinct domains. And what that means is that you can specifically target domains to have downstream morphogenesis. In this particular case, you can target one of the domains to generate these protrusions. And like why stop there? You can target multiple domains to generate multiple protrusions. So this is the idea of a synthetic embryo. So embryo also undergoes domain formation during its earlier phases of development. But here what we have is that like we can actually target the domains and get different behaviors grow out of it. This is again diving a bit more into how that happens. So we actually start with a quite simple morphogenetic function of differential adhesion. So this is the first code, if you will, in this sort of synthetic program we put into the cells. And the first line of the code is telling to these now engineered cells. So again, this was an innocent wild type cell. We put the synthetic DNA in it, and now the multicellular sort of group that cell created has the ability to read and execute. I'm anthropomorphizing a little bit just to drive the point home, but read and execute this stepwise program. So the first line in the program is telling us to drive differential adhesion between these cells. So we have cells in the middle and cells on the surface. And what that will enable us in the next step to specifically target the cells on the surface and have them undergo the string pattern. So here I'm just explaining at a high level how this would work with the genetic circuit approach, the traditional approach, as I mentioned in the beginning. So you would have cells only on the surface to undergo a Turing patterning and elect a leader. And then from that point on, you would run a bandpass algorithm to create 3 different steps. And the next slide, I'm going to actually dive into one of these to see how you can accomplish this with genes. But just at a high level, this is the choreography. And then what that now enables you, this domain formation, is to specifically target cells in the middle band, have them undergo another Turing patterning and another band pass. So that way you are getting to the 6-domain spheroid. And then from there, you target whichever domain you want to trigger the protrusion formation. And you have a clock running in the background organizing all of this. This is something we did for DARPA, which is like very ambitious, to run this type of a synthetic genetic algorithm to create a synthetic embryo. And just to, again, dive more into one of these functions. So let's talk about synthetic, like Turing patterning, how this would work with actual genes. So Turing patterning is essentially a theory set forth by Alan Turing. This is only biological work, everything else is in computation, where he actually described a very simple topological interaction between 2 agents, 2 species, 2 chemical species, that would yield the complex patterns we observe in nature in the course of morphogenesis. So these are photographs and I don't believe, okay, my apologies, I have not cited this paper, but we'll make sure to include that in the edited version. This is a paper where they captured sort of real life photographs of different patterns in nature, and then they have also simulated based on Turing patterning by changing the reaction diffusion parameters to show that what Alan Turing postulated as a theory actually could explain the basis of morphogenesis. And the paper, Turing's paper, is chemical basis of morphogenesis. So in our circuit, for example, we deploy this approach to run that Turing patterning on the surface cells to generate the initial symmetry break, the leader selection. And a little bit more specifically, again, I'm trying to take you from this like big picture of multicellular morphogenesis to how this would actually work in the cells. So Turing talked about this activator and repressor species and this topological interaction between the two. So essentially what you would do as a sort of like in the traditional synthetic biology sense, you would look at your toolbox and pick the species in your toolbox that could deliver you this behavior. So the specific genes for this we use SynMatch and Thornton, and we pick these is because SynMatch is short range. So Turing's postulation is that activator needs to be short range and the repressor molecule needs to be long range. So Thornton is a diffusible molecule and SynMatch is a short range. I'm sure you guys like heard of these or in work with these. And then you would have your, okay, so this is where we're creating that synthetic circuit. Instead of, again, transistors, we're using individual genes and wiring them up. So even sort of a takeaway from this whole, and by the way, just to give you a image for how this is looking in the dish. So you have these surface molecules of your SynMatch communicating in a short range manner, activating itself. So SynMatch is composed of a surface ligand and a receptor. So when the 2 cells connect, the activator, SynMatch, gets activated, and then not only then goes on and activates its repressor, Thornton, so the purple genes here, but also goes on and reactivates itself. So by activating TTA, which is a SynMatch activator, you're essentially recreating this. So we talked about that kind of high level. This is not exactly Bloom logic, it's more continuous, but you can still see how you can create this high level behavior of interest with genes by wiring them up in specific ways, which is the really backbone of how synthetic biology unfolds. And then the hypothesis is that once you wire up your cells this way, you would be able to, once you let them free in the cell, in the dish, they would be able to trigger that autonomous patterning. It turns out it's much more kind of, so this is amazing. Going back to the big picture, of this sort of wiring up different morphogenetic functions. And we've just looked at how Turing patterning would work in theory and wiring them up together and driving the synthetic patterning. In theory, this is great. And definitely this field will like one day gain this level of control over synthetic morphogenesis. But one thing I've learned during this project and during my time with the synthetic circuit approach is that there are just a lot of variables for something as, I'm not going to call it simple, but this is now a 6 transcriptional unit circuit like that. For the big patterning I've showed, we would need like dozens, but even for a 6 transcriptional unit, this actually proved to be quite challenging, adjusting the rates between all these proteins to make sure everything happens in the exact same time so you get your pattern. Overall, I believe my take from the circuit approach has been, which I can summarize here, is that it's great. And in theory, it's really makes a lot of sense, gives a lot of control over the tissue patterning, but it is still quite patterning as a field. It's still quite challenging as a field. We're just really in our infancy. So for that project, Big Synthetic, we have been able to make parts of it work, but just it's, I don't think to get to this level of control where you can generate any type of protrusion, probably another like 20, 30 years, we're going to be needed. And I love this approach, but that whole experience made me think about, okay, so this is a traditional approach, right? You have your wild type cell, it has these rules. We have this view of cells with sort of machines executing a embodied code. And then you can add in a new line of code with your synthetic circuit, as I've just explained. And then you have your synthetic morphology. So from wild type genome, you now switch to synthetic genome because you have a new piece of gene there or multiple genes. And then the goal is to reprogram this wild type morphology to a synthetic morphology. And again, one of my favorite examples from Wendell Lim's lab, this 3-layer patterning. Again, this is great on practice, but it has just a lot of challenges. And sort of maybe I should name this talk as challenges and opportunities, because there is a lot of opportunities, as I opened with, but I want to talk about some challenges too. So some issues with the genetic circuit approach to synthetic morphogenesis is lack of complex patterning. So we have really been able to accomplish like pre-patterns only, domains, bullseyes, and then it's difficult enough to create the patterns. If you want to add functionality, say create a specific material in this particular domain, it's still also quite challenging. And large-scale patterning too, although with the diffusible morphogens they've showed, like I showed, like a couple centimeters from Wendell Lim's lab, still you wouldn't be able to do something much larger than that. And then everything really boils down to the like toolbox being limited. So that's the sort of point I was trying to show with the Turing patterning circuit. Like you can have even in a 6 transcription circuit, you can have all your individual genes, but toolbox really need to be not only developed, but also the tools behave differently from chassis to chassis. And then delivery issues, some cells do spit out the synthetic circuits. They do not like to be engineered in that way if it's going to create a lot of metabolic burden. And then you also have payload size limits. So again, even a 6 transcriptional unit circuit, things were challenging. And they're trying to scale this up, but currently the number of genes you can introduce is limited. And yeah, chassis rejection I already mentioned. So these are some of the challenges that accompany the field of synthetic morphogenesis with its traditional approach. Again, traditional quotation marks, because synthetic biology has been around for like 25 years now. But these are some of the challenges. And through that experience, I started thinking, okay, is there another way? Nature is quite good at creating all these different structures, and we do need some control over it for sure if you're trying to get it to go to a target morphology, but we don't need to edit every single thing in the system. So then the question really became, this is a traditional approach. What about, instead of trying to control everything in the system with the exogenous circuits, instead we leverage what's already there? So what does that mean? So this is an alternative approach to synthetic morphogenesis. And this is going to be the sort of next example I'll illustrate with. So again, in the traditional method, we were saying wild type genome, wild type morphology. and add a new function and make it synthetic. So what if instead you took that wild type genome, go with the hypothesis that there actually exists rules that could drive the synthetic morphology of interest, but that they're commented out. So they're latent. And can you activate them? So without delivering any external genes or any payloads, anything exogenously, can you just give the system some non-default environmental input and uncomment that code so that you're driving this complex large-scale morphology with function? So then that's what I do to try to accomplish in my PhD. So now this is in Mike Levin's lab at Tufts and Harvard Bees. And what this was a quite interesting project. Really, the conversation started with, and I should have had their photo here. I might have them downstream, but xenobots, so the frog bots that have cilia on the surface that swim beautifully. You might have heard about them. The conversation was started, can we make mammalian versions of them? So now we have this target morphology, multicellular, and we have, so this is my spoiler alert, you have a spheroid, you have cilia on the surface, everything is mammalian, and the cilia cells are able to move. But using that traditional circuit approach, getting to something like this would basically be it is impossible right now, because let alone complex patterning, let's say you got the patterning right, to initiate ciliagenesis, which is a process we don't even 100% understand how it happens in nature, would just require a really massive payload and a like really fine-tuned orchestration, which again, as I've explained, we're just not there yet. So then really the question, okay, how do we do this? Like, how do we accomplish this if the circuit method is not going to work? Via morphogenetic plasticity. So a little bit background on this. Morphogenetic plasticity says that in the developmental landscape, where you start as a single cell, you actually have multiple routes that you can take. But that evolution, for its own selfish reasons, forced the cell to take this one particular route. So that is our default morphology, right? Let's say this route. That is our default morphology. Okay, great. I do have the paper name here. So this is Callum Durant's work in the Levin lab, where she showed that by taking a flat worm that has the default morphology and by introducing, so no new genes, no circuits, but by just changing the electric patterning in this worm, so you can cut it in half, change the patterning at the wound site, and then I encourage you to watch Mike's talks on this. The details are really quite amazing. You can actually generate a completely new morphology, right? So this is now a synthetic morphology with the wild type genome, where the system was pushed into an alternative developmental path and ended up developing two heads. So you can do the same thing and develop two tails, right? Cut in half, change the pre-pattern at the head site, and then to that of tail, and then now you have a tail. And turns out you can actually even do quadruple heads, which is quite crazy. This is the general approach to synthetic morphogenesis via morphogenetic plasticity. Or rather, this is this general exploration of morphogenetic plasticity. With non-default environmental inputs, you generate synthetic morphology. What I really became interested in is asking the question, okay, great, can we use this for synthetic morphogenesis? Can we leverage morphogenetic plasticity to create new architectures by design? Right here, you're just doing random things to the system and seeing what happens and mapping the morphogenetic landscape. Well, how would it work if you were actually trying to push it to a specific outcome? And again, reminder, particular outcome I was trying to push it to is multicellular spheroid with cilia on the surface, mammalian cells. Oh, great. Okay, I do have the photo. So these are xenobots built by Blackiston. They are spheroid, motile, multicellular, grown from amphibian embryos, and they are sculpted. So what we wanted to do, our target design specs, again, spheroid, motile, so everything is the same, except this time we want them mammalian, and we can't use embryos, human embryos, right? And then this is my sort of pet peeve. I really want them to be self-constructing to show that we can bring that quality unique to nature to the process of design. Quickly, what we did here is we took so again, in this approach, the really first question you ask yourself, okay, I want to generate a multicellular spheroid that has cilia on the surface. Instead of starting from a blank cell and putting in all the genes as a circuit to try to get there, which is quite challenging for all the reasons I explained, can I start from a cell that already knows how to do this? Can I start from a cell that already knows how to develop cilia? So with that, we've looked at, and not just cilia, but mammalian cilia, ideally human cilia. So we looked at multiple different ciliated tissues in the cell and picked area epithelial cells to start our exploration. And after trying a lot of different approaches, what really worked for us is to take these cells that live at the really bottom of human area epithelia. So these basal cells have the ability to continually generate these ciliated cells. And normally, what they do in culture, when you culture them in a matrix-like environment, they create these, so after 14 days of being cultured in a matrix, they create these cilia in structures, right? These are cilia area organoids. You might have known of them. And per our approach to create synthetic morphology by giving non-natural out inputs, we've actually treated these area organoids in a way that they wouldn't necessarily expect. So we've dissolved this matrix from the growth environment and started bombarding them with retinoic acid, which is really important in spherogenesis. And that way, we were able to get these spheroids to essentially flip inside out and now expose the cilia on the surface. So this is how they grow in 14 days. On day 0, after dissolution from a matrix here, they're not motile, nothing is happening. As you can see, cilia is inside. But after 7 days, once we put them into a non-active environment and bombarded with retinoic acid, they actually localize the cilia to the surface and start moving. And this is how cilia is showing up on the surface. And because we're here, we're leveraging self-construction, we can easily actually create a swarm. This was quite exciting. Again, doing something like this, it's not a circuit approach would just really not be feasible or would take 20 years, but I was able to get this to work within my year and a half in the lab. And that was really exciting. And now that, okay, great. Now we've accomplished our goal. What next? What are some of the drawbacks of this approach compared to the circuit approach, right? Was able to get this complex morphology and behavior in a quite short, hit all the design specs, so to speak. Default morphology was for these cells to create airway cells, lung cells, but we were able to push it to this kind of alternative path. But now what happens is we're getting ciliated spheroids, but this is also what we're seeing. There's a massive variability, and not only massive variability in morphology, but massive variability in behavior. What then really happens in this leveraging morphogenetic plasticity approach is I'm learning that you get your target architecture, but there's no free lunch. You are also getting a massive variability in your output, which actually could be interesting for developmental biology and engineering. If you always want to get your particular target spec every single time, I think more optimization is needed. But for developmental biology, for exploring whether the rules of morphogenesis in nature also applies to the process of synthetic morphogenesis, this was actually very exciting and interesting. And to dive a bit more into that, we've asked the question, could there be character formation here? All of these guys pretty much look the same, kind of variation on a theme, which is a very known thing in nature, like fingerprints, right? They all look similar, but none of them the same, very much like Anthrobots. And also, and same behavior phenomenon is seen in butterfly wings, like snowflakes, fingerprints here. I don't have it here, but also shark teeth. So just turns out there's this whole kind of field of character formation, characterization in developmental biology. And also, I came across this really cool thing. So it turns out FBI actually has this way of characterizing human fingerprints into 8 different categories. So started looking at the Anthrobots and this process through these lenses. We asked the question, okay, can we detect some sort of character formation here? So I usually in my talks don't go into the stats and all of that too much. So I'll just tell you just of it, and in Q&A we can go back and discuss the specific methods. But essentially, we have done 200 plus with 200 plus time-lapse videos of these bots, and then ran it on a simple 2 matrix unsupervised clusterization along the metrics of gyration, like how much they go in circles, and straightness, how much they go in straight lines. And discovered that actually there are 4 different categories of Anthrobots movement. They can only go in circles or in linears or make these arches. And then you also have this 4th eclectics category where they have different behaviors. They switch it unstable. These are more stable categories, but there's a 4th unstable category as well. And here are some examples. So this is a circular bot, and it also sometimes changes direction. So that's something to actually further look into, like chirality. And then here's an example for a linear one. So it turns out, yes, there is massive variability, but they also self-organize into discrete movement types. And I can go more into, so there's some switching between types. So the most stable category is actually type 1, the circulars. Like once you're circular, you're like circular for life almost most of the time. Type 2 is a little bit less stable, which is linear. Type 3 is less, and then type 4 expectedly is the most unstable. Only almost 40% of type 4 bots stay type 4, otherwise they resolve into different categories, which again makes sense because those are just non-stable. This is more like a transitory state between actually stable points. Okay, great. So now we have our sort of behavioral character formation, right? And we see this in the dish, quite exciting. What about morphology? Is there a similar character formation in morphology, given looking at this diversity? So this was a bit more challenging to analyze because this time you have more, you can't just say circular, linear. Morphology is more complex to analyze. Again, happy to come back to the slide and talk about how we pick these indices, but these are essentially different ways to talk about morphology and structure. Similar approach, not this time we have a multi-axis PCA instead of two-axis PCA, but again, with clustering, unsupervised clustering, we're able to identify these statistically distinct clusters at the level of morphology. And turns out you have 3 different morphotypes. You have fully ciliated ones, small, and then you have bigger patchy ones, and then you have middle-sized polarized ones in terms of cilia distribution. Again, more things we can come back to. And with the hierarchical clustering, actually, we've also been able to identify what is the first decision point in this developmental path. So you first actually decide as an anthrobot on your size and shape regularity. Am I going to be a bigger bot or a smaller bot? If you're a smaller bot, you're actually locked in place in terms of your morphotype. But if you're a large and irregular, you can actually either be non-polarized, like here, then you have morphotype 2, or you can be polarized like here. So there seems to be that like decision tree that I showed in the first place in the exploration of the latent landscape for morphology. So great. So now we have our 3 sort of these idealized morphologies: polarized, patchy, polycellular, and we also have our, so I took out eclectics here because that's a transition state. We also have our 3 different motility types. Really, then the next obvious question is there mapping between the two? Does morphology drive behavior? Which is another phenomenon we know in like natural morphogenesis, and it would be quite exciting to see that happening in, at least asking the question, in synthetic morphogenesis. Through basically overlapping these 2 analyses, we have also seen a statistically significant correlation. This doesn't prove causation, but significant correlation between different movement types. So the ones that wiggle in place and the ones that have full cilia, ones that go in linear patterns that I showed earlier, and ones that have patchy distribution of cilia, and ones that go in circles, and ones that go in have polarized cilia distribution, a significant correlation. Again, a really exciting finding that morphology seems to be driving, at least we have a strong indication of that in synthetic morphogenesis facilitated with leveraging plasticity. So yeah, essentially what that enables us is to map their Wellington landscape, right? So Wellington landscape, the idea of single cell growing into different fates, and seems to be based on different checkpoints that an anthrobot initial cell goes to, it can have different outcomes. So we did get to this target morphology, but looks like when you do that, you need to actually look closer and realize that the target morphology can further branch out. So that's what we learned in this process. And couple quick fun things about Anthrobots. Turns out when you aggregate them together, sorry, I lost my slides. Should I reshare?
Yeah, yeah. It was so interesting. Please do.
Okay. I don't know if it's like a Zoom thing, but let me see.
They just suddenly disappeared, or
Zoom said screen sharing stopped. I don't know. Okay, let's see. So I'll actually continue telling about what we did with Anthrobots in the meantime. So the really cool thing there was we've tried to see, okay, so all these amazing things about Anthrobots as individuals, but so what happens when you bring multiple of these guys together? Is there any type of like collective behavior that transcends what a single Anthrobot can do? And that's, okay, here we go. That's what the answer seems to be yes. And just want to share a couple insights for that as well. Share. Okay. Can you guys see my screen again? Okay, great. Again, so going back to the initial actual reason for why we want these guys to be like mammalian, we wanted to, and I guess I didn't clarify it at the time, but we want to use these for potential therapeutic applications. Can we put them into different tissues? So these are some of the aorta, jugular, and like bigger sort of tubes, essentially. We've extracted from rats just to kind of like try it. Can we put Anthrobots in there? Again, very optimistic. So it didn't work great. There are a lot of challenges with that, which we are like separately needing to solve. So what we did is, okay, let's take a step back and look at a less ambitious model. So this is just a 2D layer of neurons with a scratch in the middle. So not of like fancy 3D ex vivo tissue, but a culture, neural culture in vitro. So turns out Anthrobots can actually not only move around in a dish, like I showed, but they can also move across these lesions. Again, it's not like a full-on tube, but still has a similar topography. And different motility patterns actually yield different behaviors of exploration. Again, morphizing a little bit, but different ways to cross it. And we've characterized actually how that would look like. And based on what your goal is in deploying the tissue, Anthrobot tissue, it turns out you actually want to pick different motility types. And so we'll get to that. But another thing we've discovered is that, yeah, like when you put multiple of these guys together, they can also fuse into larger structures. And the larger structures are still motile. So you see like a large guy pushing the small one out of the way. Bad news, when you put them into tubes or lesions, they're too big to really do that kind of not exploration, but let's call it traversal. Even though they have like cilia beating like crazy, they don't really move. But then what we've discovered in doing that, in the course of 3 days, their just presence on this lesion acts as some sort of a bridge and helps this tissue underneath it heal. So this was a bit of my kind of architect side, trying to see if I can get larger structures by aggregation. Turned out this gave us this real cool thing in therapeutics where super early indication, this really a lot more research is needed on this, but super early indication that they might be able to help nerves knit across. Just quick controls, then we put agros slabs instead of bots, no healing, and you can then remove the bot and then the healing space. And then the healing, by the way, tissue quality in the healing site, so site number 1 is statistically indistinguishable from the original tissue. All right, so yeah, a lot more research is needed to use this in neurodegeneration, but it is very exciting direction. So next, like the question, can we bring the two together, right? Can we start putting now exogenous genetic circuits to these Anthrobots or similar kind of biobots that we grew by leveraging morphogenetic plasticity to really bring the best of both worlds in synthetic morphogenesis and transcend our current limitations due to technical difficulties in traditional synthetic biology. All right, with that, I just want to thank everyone at Tufts. I didn't put my MIT people here. Yeah, sorry. So Ron Weiss was my advisor at MIT, and my project teammates were Jesse Tordoff and Catherine Kirimaji in the MIT project. And at Tufts, big thanks to my students, Pranjal, Hannah, Ben, other Ben, and advice from Simon Garnier on all the statistical analyses. My advisor, Mike Levin, and committee members, Kelly McLaughlin and Barrett Trimmer, and my external committee member, who's the inventor of the Nudge system, by the way. So with that, I'll stop. I think I'm a couple minutes over my 45-minute mark, but would love to chat more about via Q&A if you have any specific questions.
Fantastic. Thank you so much. That was really wild, honestly. And I had no idea that you moved basically between fields and landed there. What a wild career journey as well. It's absolutely crazy.
Yeah.
Thank you. It was a really great talk. If anyone has questions, feel free to raise your hands now. Then I'm just going to take them hand by hand. And if you can't raise your hand but have a question and want me to ask it, feel free to drop it in the chat and I will get going on. I think you stumped a lot of people here. Usually, we have one of them already from Marina. I don't know if you can unmute, but I will just ask it for now and you can follow up, Marina, which is a quick question. How did you eliminate them?
Oh, the bots. So that is actually the next paper that will be coming out in the next few months. But yeah, that's an excellent question. If you want to put them into tissues, you don't want these bots to be just never dying and becoming like zombies and potentially causing crazy tumors. No, so turns out every single Anthrobot degrades eventually. Some degrade faster than others, and we're characterizing that right now, but every single one essentially degrades into individual debris. I don't think I have a video of that here, but just, yeah, stay tuned. Next Anthrobots paper from me and Mike will specifically tackle that.
Fantastic. And if you could perhaps speculate, this is already, I think, what makes people in this group in general so excited about this type of research is that it's A, so new, and then B, that the application space is almost unlimited to some extent. It has so many implications across a variety of different fields where people before that didn't even know how they were connected. So could you speculate a little bit about possible implications for cross different fields in the next 5 years? If everything goes right and you're like, all these experiments are successful, then how would it influence? What would other fields need to pay attention to, basically? What's coming on the line for them?
Yeah, I know. Another excellent question. So one thing I should talk a lot about, and I guess I didn't unpack that in this talk, is that like this approach, synthetic morphogenesis, has a lot of implications and applications in diverse fields. So for engineering, obviously, if we can get a greater control over engineering synthetic biological tissues, that would not only potentially unlock this sort of level of, okay, now we have this new fabrication modality, and we don't need to keep using the old sort of high carbon, high cost approaches, but that we can do more sustainable construction faster by leveraging this sort of like self-replication aspect. So in that sense, I think for architecture and construction industry, there is amazing potential. One thing I keep seeing architects or engineers who are interested in biodesign over focus on this material forcing, just swap out the more toxic raw materials with biological ones, but then keep everything else the same. Go on to 3D print, mold, cast, sculpt with that material to essentially like keep on embracing the traditional way of building. But that's why I like this slide a lot. I want to show here we can do a lot more than that. Let's not stop at using biogenic materials, but let's actually dive into the material, to the morphogenetic code, and edit that to get that material to build itself to the new architecture. In that sense, that's super exciting. But also not just for architecture and like building things by design, also for organoids, right? Or just organs in general. Like if we get greater control over morphology of these tissues, we would be able to, and that's the motivation behind the synthetic morphogenesis field in general, we can create new organs from patients' own cells, and really the organ list, wait list would be like thing of the past. Also, just other developmental diseases or even cancer. Those are all cancers are inherently architectural problem, right? Just tissue deviating from what it was supposed to do originally per its morphogenetic code. Now it's doing something different, and like how can we restore that? Obviously, a lot of potential for medicine too. And then for developmental biology, as I mentioned, like just when you push the cells from what they are conditioned by evolution to do into these like new morphologies, we're really exploring more about how the architectures, like natural architectures, emerge on our planet in the first place. What are the rules of morphogenesis and evolution? Evo-devo questions. So I would say those are the main ones. I can keep going, but yeah, those are the main exciting directions.
That will keep some busy, and for some people busy
Yeah.
for a while, I think. I know that we only have a little bit of time left. We have 2 hands raised, so I'm just going to go with Micah and then David, and we'll see how far we get.
Along with genetic drivers and bioelectric network drivers leading to morphogenesis, there are also mechanical stressors that can lead to different morphogenesis, right? So like your bones grow in different directions depending on where the pressures are and stuff like that. Have you thought about how to leverage this in a synthetic way at all, or do you know anyone who has?
Yeah, no, that's, I guess I have all the like best questions. So yes, that's why actually be like, I'm looking for the slide that I really want to plot for this question. That's why I actually like to call it morphogenetic code as opposed to the genetic code, because like you said, in addition to the genetic drivers, there are all these like external drivers. And so morphogenetic code is really the genetic code plus the environment, plus all the sort of environmental impact points that you're talking about. And that's why I think Anthrobots are like really powerful, because it is like precisely our changing matrix, sort of this surrounding matrix's phase is what drove this eversion phenomenon going from cilia inside to cilia out. So by removing that force, we were able to, and with retinoic acid, encourage cells to undergo like real large scale tissue reorganization, like eversion. Yeah, I think that is a really important like morphogenetic knob that we can tune. And I think there's hyperfixation on genetic level, but that kind of physical, like physiological even level can unlock so much. So yeah, and a lot of people are working on that for sure, like synthetic physiology.
Awesome. David.
So I was just curious to hear you talk about the how much bang for your buck you can get from engineering, like the internals versus the externals. Because I think about one of the reasons why we have to source raw materials is because when you go build something in our world, those raw materials aren't just right there naturally in the environment. They're like in all different parts of the world. But in the cellular environment, it's been evolved to be able to grow in the concentrations of things that are immediately around it. And yeah, how far do you think you could get from like engineering just if you give it the right stuff around it as opposed to give it the right stuff inside? Like, how do you think about the how much you can get from the two strategies?
Yeah, so this kind of like relates to the previous question, right? I would say that both approaches have limitations and opportunities, and that's what I actually try to like really drive home in the talk. I would say max out them both. It depends on what you're trying to build. If it's something like simple, instead of going and searching the latent space to find a cell type that might already have it in it, maybe engineering it via a circuit is easier. But if you want to build something more complex, like in the case of Anthrobots, it was definitely worth it to do that screen. But then that also has limits and coding the genes. So that's why I actually like to like always, this is very simple, right? We haven't put in anything fancy, just the fluorescent protein. But bringing genetic circuits into picture to do then more. So maybe engineering some communications between these bots or getting bots to release something into the environment. Like that would be so difficult after having had the Anthrobots to now, without any engineering to get these guys to release a particular like small molecule, let's say. It would be really difficult if you just wanted to go and search for the latent space for that. But you can now bring in the circuit approach and just put like an inducible promoter downstream of that, the small molecule of interest, and then get that, or protein, whatever you're trying morphogen, get that be deployed in that way makes more sense. So it's really, my point is that one is not better than the other, but more that bringing the two together will really, it's going to be like 1 + 1 = 3+. So that's really going to drive the ambitious like goal of synthetic morphogenesis. Did that help?
Yeah, that's great.
Yeah, it's a little bit like context dependent.
I know we're at time, but if I can sneak in one more question, which is just that
Sure.
if people are really excited about your work, what can they do to help support? Are you looking for any collaborators, any funding for specific areas, anything at all that people in this group can do to help you work along?
Yeah, no, thank you for that question. So I'm actually really interested in, so yeah, absolutely reach out. I will, Allison has my email, so feel free to distribute that. Allison, totally feel free to reach out. I think one of the things I would love to see is people trying this approach like in their own labs. Think about what synthetic thing you would want to build and then go looking for a tissue and like exploring a little bit. Like if this really inspired people to create their own bots, like that would be the really, I think, ultimate success. And more specifically, like where I really want to branch out to is maybe actually looking into plants. Can we bring this type of synthetic morphogenesis? Everything I showed was in mammalian cells and a little bit bacteria, but because that's where the toolbox is rich. But I think going into plants will be really exciting because now we can unlock all kinds of new materials. And yeah, I'm always looking for funding, of course, but specifically if anyone had funding sources they're aware of for plants, it's less popular than mammalian realm, but that's something also, yeah, I'm looking for. But any grants in general, grants are always welcome.
Okay, amazing. Thank you so much for taking the time to join us. That was a fantastic presentation. I'm really, it's such an exciting field that's exploding into our face right now. So thank you. And thanks everyone for joining. Have a good one.
Thanks for having me. Thanks for great questions.